From Microbe TV, this is Matters Microbial, a podcast about microbiology, microbiologists, and microbial centrism. This episode was recorded on November 13th, 2023. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 16th episode of Matters Microbial, and I appreciate every viewer, every listener, and every commenter. Thank you all so much. I'm hoping to have a little less of yours truly at the beginning of these podcasts and get more quickly to our podcast guests. Still, I do want to introduce one denizen of my Wunderkammer. It's a wonderful tiny sculpture of a bacterium complete with pili, wielding a micropipetter such as I use in the lab every day. It was made by an artist who goes by Chocolate Menagerie, and I will link to their Etsy shop in the show notes. During the last few episodes, we've discussed the microbiome a little bit. The idea of the microbial partners that have co-evolved with us over time. And not just us, but all living things, pretty much. As always in biology, we often learn things applicable to humans from other and often simpler systems. Thus, I'm very pleased to introduce Dr. Nichole Broderick of the Department of Biology at Johns Hopkins University in Baltimore, Maryland, to tell us what her lab group has uncovered about the microbiome of fruit flies, smurf flies, and how all of that might be important to human beings. Welcome to our Quality Quorum, Nichole. It's so lovely to see you.
It's great seeing you, Mark. Thank you for having me.
So I think the first thing that might be very useful for our listeners and viewers is a quick thumbnail sketch of how you got to where you are.
Yeah, so I grew up in the state of Wisconsin and went to the flagship public institution in my state, University of Wisconsin-Madison, and I entered very much as a first-gen student with an idea of going into pre-med. I thought genetic counseling sounded interesting, and pretty soon, though, my second year, decided to get a job in a lab because I had been told, A, that can be good on your resume, but B, that I also needed some money while I was going to school as well. And I naively, and I always stress this to students, thought that going to a lab to wash dishes would not be a way to get exposed to research. I always tell my students that's not true. It's a great way to make an entree into a lab team and show that you are interested and excited, but I did not know that. And so I managed to pick a lab that didn't have washing dishes as the top thing, and it was sorting insect specimens and helping raise insects for a fungal biology lab that was a forest entomology lab that was looking at a lot of host microbe on the insect side, but especially a lot of fungi in that case. And so that's how I started doing research, and I got hooked, and the rest is history.
Can you give a quick, again, thumbnail sketch about the overall goals of your research group?
So that intro to working with insects really, I guess, got me excited about entomology and insects as a model system. And so I continued to work in insects. I had worked in moths and butterflies when I was in grad school, and then in my postdoc transitioned to working in Drosophila, so I could have a genetic model system to be able to manipulate. But my big interest has always been trying to understand how microbes influence animal biology. And I think insects have really interesting relationships with microbes that also extend to what we know about other systems. And so they're a great tool, I think, to be able to truly understand some of the basic ways that microbes impact insect physiology and evolution.
Oh, I couldn't agree more with that. And it's interesting because some people that I speak with don't seem to understand why we would look at something like insects to learn more about, for example, the microbiome. And I happen to know your record pretty well, Nichole, so I know that you can say a few words on that subject.
Yeah, no, I think, insects are the most abundant animal on the planet, of at least macroscopic animal. And when we think about the many interactions they have with microbes, and the fact that animals, all of our eukaryotic cells evolved in the presence of microbes and bacteria. And so they're just a great system to try to tease those apart. They have really interesting ecology. They have interesting impacts on things like reproduction. And we're only at the beginnings of trying to understand. And the work that we've done and others have done has shown that a lot of what we find, in particular work like we're doing with the microbes' impact on the immune response or gut development, are really themes and pathways and microbial factors that um, have impacts on insects all the way up to what we see in humans. And so they really can both inform insects as an interesting group themselves, but also what we know about other animals.
Oh, I couldn't agree more. And it's really true when you think about it that what we learn about some of these, and I hate the word simpler, but you know what I mean, simpler
Yes.
systems can be transferable to issues involving human health and wellness. And we've seen evidence of that in many, many systems. And I have to say that the idea of using insects as a model system to study diseases, even in humans, has been fascinating beyond words. And I know lots of has been discovered about it. Parenthetically, you, Dr. Broderick, were kind enough several years ago to send me a particular type of bacterium, which is called
Yes.
Serratia. It's actually a subspecies of it. And if you look at this picture, it makes this beautiful red pigment called prodigiosin. And you told me that it would actually kill insects that consumed it. And as you can see, we call that the Red Death in this photograph here. But it isn't anywhere near as cool as some of the things that you've discovered. The important thing for our listeners and viewers to remember is by studying how some microbes can cause disease, even in fruit flies, it's still relevant to what happens in mammals. A great deal of work has been done of that in many labs all across the country.
Yeah, no, I mean, I think we have two recent studies that I think sort of are really good evidence of that and support that theme. So my lab very broadly looks at a lot of aspects of insect physiology, but we also use the gut to try to understand microbe-microbe interactions within an environment. And if you think about the gut, it has entry and exit points, but it is kind of a closed system. So you can try to understand sort of what are the dynamics that happen with interactions. And it's something that a number of teams have also taken to their advantage in understanding that. But I had a student, Alex Barron, who just graduated and recently in October, who did some really cool work where we've been looking at how the microbiome, and in particular, certain members of the microbiome, lactobacillus, lactoplantibacillus plantarum. They just recently changed the name. Anyone who's a taxonomist or who suffers at the hands of taxonomists will understand the issue there. We'll call it LP for the rest of this. I'm still getting used to it. We found that it, like many lactic acid producing bacteria, which are important with a lot of food fermentation, things that we love to eat and drink, kombucha, kimchi, yogurt, that just like in those systems in the Drosophila gut, that this bacterium acidifies not only the gut, but also the food that the fly is feeding on, and also the other substrates it comes into contact with. And through Alex's work, we were able to show that what's really intriguing about this is another member of the community. I think I have a picture of our acid interaction assay, where we found another member of the community, an Acidibacter, an acetic acid bacterium, actually quenches this activity. And so what's intriguing about this is depending on their interaction, it either inhibits or is more permissible to different types of invasive microbes entering that niche. And in particular, we looked at some gram-negative microbes, an E. coli Nichole that's actually a human probiotic, um, a and then two fly-specific strains, and Pectobacterium keratabra, um, and a that's non-lethal but causes a lot of damage to the gut, and the fly is able to recover from, and then a Pseudomonas entomophila, which we know is a lethal pathogen of flies. And so the lactobacillus acidifies both the food and the gut and reduces the population levels of these invasive microbes. And so we think this is really a great example of a mechanism where we see things like in the human vaginal tract or other aspects where pH is really important, even though it's such a simple thing and something we've understood for a long time, can have really big impacts on sort of not only the way microbes are interacting, but how the microbiome itself is shaped and built and functions.
That's very interesting you would bring this up, Nichole. It's true that lactic acid bacteria are important in human health, as you've mentioned, and absolutely important in a number of types of foods of which I am fond. But I would also like to say that many of those make, and I believe they're as a class called lantibiotics, they make antimicrobial compounds of one form or another. Is there any evidence that this denizen of the Drosophila gut is doing the same thing?
So that's the next part that we're trying to understand. The pH was an obvious one. Initially, we were really excited, thinking like, well, maybe it's a bactericin, or maybe it's an antibiotic, a new antibiotic we could study. That relates to the course that I love to teach. And initially, we were a little bit, and he did such a careful example of trying to of being able to show it really is pH. And our first reaction was a little bit like, wah, wah. But ultimately, what we realized is that it's a really underappreciated mechanism that microbes can use to kind of carve out their niche. And we think it also has really interesting impacts on the microbe, the fly itself, and also on other microbes in the system. But we have evidence for certain that there's other things that they're being produced. And we're expanding this now to also, that was only a two-way interaction, is what happens when we start to make these interactions more complex, or if we bring in different microbes. So you can imagine, depending on the player there and what they're producing, the metabolites they're producing, it could really shift sort of what's happening with these interactions.
It's very interesting. It is absolutely true that that low pH idea, acidifying an area, has a kind of a restrictive effect on many. It's behind one of the things that you're doing when you're making sauerkraut, of course.
Yep.
But as you mentioned, it is something found in the human vaginal tract that can help reduce the incidence of bacterial vaginosis. I was also unaware that imbalances in that particular region can cause greater sensitivity to certain sexually transmitted diseases. So, this is a really big thing in a really simple system when you think about it, where it's tractable, as you've mentioned, on both sides.
Yes, no, and that's a thing we find really attractive about it. Also, because we can make flies azenic, we can eliminate their microbiome. It's very easy in our case. We just have to use household bleach on eggs and the embryos, and then we just transfer them into sterile environments with sterile food. And as long as they have the right nutrients available, we can raise them without a microbiome. And that allows us to put back specific members and look at some of these interactions. But even without doing that, one of the nice things about our system is that we only have 5s to 10s of different types of microbes in that environment. And so we actually can ask even more complex interaction questions without having to sort of artificially reduce the complexity of the system. And what's really cool is that we see the same biology happening. So it means you don't need thousands of species in the human gut to see some of the same impacts.
And that's really critical to emphasize for the viewers and listeners is the human gut microbiome is like the Brazilian rainforest of microbiomes. And you have, as you mentioned, did you say 5 or 10?
We get, 10s. This is the best way how I like to say it. Because if you go out in the wild, you might find 20, but yeah.
And that's something else I wanted to mention to you. It's absolutely true that we are a tube, where food comes in at one end and it exits at the other, putting it delicately. And I've often been interested in the ways that we're able to restrict what takes up residence in our gut. I make jokes about things that are just kind of passing by that you brought in with your food. And then there are some things that can set up shop, call them, for lack of a better term, and I apologize for the pun, squatters.
Mm-hmm.
And this business of how you maintain a particular population is fascinating. What I'm hearing from you is a mechanism to maintain a particular population. And there's evidence of that, of course, in the human microbiome, but it's much more complex.
Yeah. No, exactly. And I think, we think pH is just sort of the first layer of that we've found. And, looking at this with other interactions will help us understand other rules to sort of how things establish, how they structure, and then how they function together. Yeah.
I like how accessible that is as a concept, because this idea of the low pH, the acidic conditions, is something that makes sense to a lot of people.
Mm-hmm.
When I have students that have trouble understanding the barriers to microbial, I hate the term invasion, but you know what I mean.
Yeah.
And I would point out that our very tears have antimicrobial properties, to the point where I had a student who wanted to collect her own tears and test it for antimicrobial activity. And no, it wasn't by taking tests.
I
It
was
was
just going
really
to
fun.
say.
Yeah, she would cut up an onion, like being interviewed by like some of these people in the 70s that were trying to make people cry. It's actually pretty amazing. That's a wonderful story so far.
Yeah.
Are
So
you,
that's one that we're quite excited about. And sort of students building off that to understand some additional players that may be involved with those sorts of rules.
and this work was done primarily by, am I correct, an undergraduate?
No, this was a PhD student in this case, but
Wonderful.
with some undergraduate researchers, both at the University of Connecticut when I was there, and then continuing now at Johns Hopkins.
Wonderful.
Yeah.
Are you going to lead us toward a more purple way of looking at the world?
Well, yes. Another study that we're really excited about, but we're really in the thick of it and trying to wrap our heads around it. And I like it because it's actually a situation where the microbiome is not important at all. And that's with a new pathogen that we're working with, which is called Chromobacterium subtigae. And it's this beautiful, charismatic microbe. It's got this gorgeous purple color because it's known to produce violacin, which does have it's a metabolite that has impacts on different cells and stuff like that. And we became interested in it as a new model because it's also known to have some insecticidal activity. It's actually a registered organic insecticide, which kind of takes me back to where I began my PhD work with working with a different organic insecticide, Bt toxin. But I had a student who really came in, was a microcentric student, and she was like, I flies are fine. I know I need a host, but I really want to work on microbes. And I was like, well, I have this interesting pathogen that we know kills flies, but we have no idea why. And it's been just a lot of fun to try to understand this and to watch the student, Madison Clark, who will be defending her thesis in January, just get us at a point of understanding sort of this timeline of death. Because one of the interesting things about the pathogen, and I think that was in the graph that may have gone up with the purple color, is we know that a white strain that we have that doesn't produce this purple color isn't as lethal. But the time to death takes quite a long time. They don't start dying until about 6 days after ingesting this bacterium. But what's fascinating is as soon as 4 days after that, we can't actually culture the microbe anymore from the fly. But if you look at signs of damage prior to that, and this is where the Smurf flies come in, we see that as early as a day or two after ingestion of this pathogen, they have these super leaky guts. And so we do this by basically putting the flies onto blue food dye. And you can imagine if you've got holes in your gut, the blue dye goes everywhere, and we see these lovely little Smurf flies, all blue. And if it stays in the gut, and the gut is the integrity is maintained, you can just see the blue line. You can see those in the controls of just staying within the gut. you can see on this slide, we have two groups of flies. On the left are flies that we've fed the white pigmented bacteria strain, the wild type control. And on the right is the population, is some flies that we've fed the purple pigmented strain, our Delta VOS, and that kills, is more lethal. And what I hope you can see in this is that in the flies that are not exhibiting injury to the gut or gut leakiness, you can see a thin wisp of blue food through the abdomen of the fly, and that's actually where it's all within the gut. So it's staying within the gut epithelium. On the other hand, you can see that most of the flies, like 75% of the flies that had the VOS strain at this time point, they're all blue, and that's the Smurf phenotype that we say. And in that case, the damage to the gut is such that blue food dye, this is also blue food dye number 1, so it's actually in a lot of our human food, is now leaked into the rest of the body cavity and into the bloods, the hemolymph and the rest of the body cavity. And so that tells us that those flies are experiencing damage. And so you can see that not every fly that's gotten the pathogen at that time point is showing damage. We have the example of the one that we have the arrow to saying non-Smurf, but most of the flies have. And certainly after a few more days, this was a day 2 time point, almost 100%, if not 100% of the flies are showing this sort of damage phenotype. And so this is set up kind of this intriguing way of us trying to understand how are flies actually dying, because they have this damage, obviously, that's happening. Then we can't find the pathogen anymore. The microbe, Chromobacterium, is no longer there. And then 6 days later, they start dying. And so one of the things we're trying to understand is sort of what's happening at the gut level versus what's happening to communication with the rest of the body. But Madison has found out at least that there's these guts look really funky around the time they start dying. And so I think there's the image I have there of full length fly guts. You can see them with, it's staining the Zdapi stain. So it's staining the nuclei of the enterocyte cells. And I hope you can appreciate at least that the one with the VOS, the purple strain that's been fed, they're really shrunk. They're really shrunken is all we know how to say. And so now we're just at the beginning of trying to understand what's actually happening there and what's contributing to the fly death. But it's a really fun bacterium to work with, and we're just really enjoying what it's telling us, not only about how animals die. We think it is maybe sort of a sepsis mechanism, but we also are using it as a tool to really push the cell biology of the gut. So we think it's going to tell us a lot about repair mechanisms in the gut and how animals can or cannot maintain homeostasis. In this case, the fly can't maintain it. And so we're really interested in understanding why. We're really curious about why it's producing these compounds that have this effect. But you might be wondering, the audience might be wondering, well, what is the benefit to the bacterium? And that we actually don't know, because in this case, what we think is that since the bacterium has lost so much from the gut, we can't find it in the fly. What we do find is a lot of cellular debris. So now we're trying to understand, is there sort of are we having like a fly diarrhea? Is there a population of the Chromobacterium that are expelled? And then you can imagine that's a way of sort of passing yourself on or infecting, other. You think about this happening on a fruit, could infect the rest of the fruit, right? And then you can infect other microbes. Or is this just, a random, not really, maybe it's not directed. Maybe there's no benefit to the Chromobacterium. It just produces these molecules that in its in lifestyle, in the environment, benefit it in other ways. But that in this context, it's able to cause this damage. And then that's the sort of impact on the host. So we don't have a good answer. My guess, based on what we know for other environmental microbes like this, is there probably is some benefit if you think about in the soil where these microbes are, or in the environment, in water, things like that. Coming into contact with a host can offer them an opportunity to access some new nutrients, to grow, to exchange DNA with other microbes. That's another good place to do it. And then they may just pass out of the system. So these are questions we don't have answers to yet, but that we're interested to understand.
Reminds me a little bit of what I was saying, Nichole, about in kind of ingesting microbes from the environment and which just pass through and which set up short term. And that certainly we can all come up, and we've sometimes had the experience, all of us, of food poisoning where that happens. It doesn't happen all the time. It's a very interesting kind of way of looking at it because inside the gut is quite different from outside
Yeah.
and affects upon the resident microbes as well. So I think it's really fascinating. The leaky gut idea continues to really be in my head on this.
It's a great phenotype.
Do you think this is related to all the commentary I've heard about leaky guts, even in human, and especially as humans, how it might be involved, in fact, with autoimmune disorders?
Yeah, so I think there's some sort of probably loss of like whatever's leaking into the gut is sort of probably establishing kind of a loss of the fly to just be able to do other physiological functions. Like with that really shriveled gut, we're wondering like, can they even absorb nutrients anymore? Like, what's happening? And so that's where we think, in terms of thinking about sepsis models, not septicemia, but at least sepsis models where you can have maybe these, you know, the immune system kind of takes over and it kind of then offsets the balance of what the host needs to do to just maintain these other physiological functions. So we just got some RNA-seq data back. We're trying to understand that, but that's something the next student's going to have to pick up and work on because Madison's done just an amazing job to get us to this point.
Well, it's an interesting idea to me because if I'm hearing you correctly, the Chromobacterium vanishes after it's caused damage.
Yeah.
So I'm wondering if that's due to innate immunity responding to the infection.
So we think there's initially a number of mechanisms that you would want to activate if you were encountering this. So I mentioned this other pathogen, this Pectobacterium keratoverum that we work on, and that's what we consider a non-lethal pathogen because we know it causes amazing damage to the gut. The gut shrinks in length by 50% just a couple of days after eating it. But it is able to engage both the immune response, but then also the epithelial renewal response. And so it basically replenishes that gut, is able to activate the stem cells in the gut that repair damage. And so the fly recovers. And so what we see in this case is that early on, like that day 2 to 4, the flies that have fed on Chromobacter seem to be trying to do the same thing, but then something crashes. And so what we're interested in is trying to understand, what's the trigger that leads to that crash? And then also, even though I've talked about violacin, we have preliminary evidence that it's not just violacin, that there's probably some different metabolites that are induced at the same and produced at the same time. So as much as we love the purple color, we don't think it's the causative agent.
Well, I'm quite sure you've read the same studies on this, that there's a lot of evidence that violacin, for example, tends to ward off protists that would be feeding upon bacteria that make that particular pigment, number one. Number two, there's some evidence that it's actually able to protect against attack by the bacterial predator Delavibreo.
Yes.
And I, of course, have E. coli that overexpresses violacin anytime that you'd like to, put that purple doodad in there. It gets almost so dark you want to, of course, I've already mentioned, given my XY status, it's hard for me to name colors, but it's very dark indeed. But I think it's a fascinating one. My understanding of Chromobacterium in general is that there are all kinds of active molecules being made.
Oh, yeah. Yeah. And so that's also the next step is trying to do some either mutagenesis to look for loss, but then also some metabolomics to try to understand what are these microbes producing and can we kind of identify what the target, what the potential molecules might be.
Well, I think it's fascinating studies. I wouldn't be at all surprised to learn it's something to do with quorum sensing, simply because you have this initial colonization activity. And we know that when Pseudomonas aeruginosa, when I say we, I did not do this work, Pseudomonas aeruginosa infects Drosophila, certainly quorum sensing is necessary for the killing that's observed there.
Exactly. Yeah. And so we assume that it's another metabolite that's being turned on when all the other kind of quorum sensing genes are, which VIOS is one of those. And so we just happen to have a constitutively, active producer that allows us to see it earlier.
So now I'm thinking a lot about that serratia strain of yours. And I don't have the ability to analyze things that you do, but maybe it's as simple as setting up colonization and leaking into the hemolymph, I guess I would say.
Yep.
There's not really a bloodstream, but
Oh, that's
you get
exactly
the
what.
idea.
Yep.
And I don't know. I mean, your hands are going to be a lot better than mine.
Well, you know where to find me, Mark.
I do. I think a better use of our time perhaps is to tell the listener and or viewer and hopefully commenter why this would matter in terms of thinking about issues that involve humans.
Yeah, so I think it both on the sort of health promotion aspect that I described in the first sort of study that we were working on, and then this latter trying to understand sort of how perturbations caused by microbes can lead to the loss of homeostasis. We're excited to think about, can we screen for different things, drugs and stuff like that might, prevent death? Because then we might have a screening drugs, and especially unknown drugs through flies, is a lot easier to then get to potential sort of therapeutics. And so, I think we can think even that far when we know we have small molecule libraries and things like that might be a source. But I think in general, it just helps us understand sort of, what are the important microbial factors, but also animal pathways and mechanisms that allow them to either live benignly with the microbes around them or recover from disease. And so I think that's what, as a group, we approach sort of the work that we do.
I think it's fabulous work. You've already mentioned several of your, I should use the word colleagues, several of your colleagues at different levels in the scientific pathway.
Yeah.
Tell me a little bit about how you recruit students, what you look for.
So, the work that we do really is at this intersection of thinking about microbes and thinking about hosts. And so I think it in some ways, then it can be a big tent for welcoming people that are interested on specific aspects. I have students who are very host-centric. I have students who are very microbe-centric. And then I have, a lot of students that meet at the middle. And so I think being curious about biology, being a good observer, so you notice the things that are a little unusual or that don't. Sometimes our phenotypes aren't huge, like that shrunken gut. Sometimes it's a little bit more subtle. So it's trying to be a good observer of the biology and the physiology of the animal and the microbe. And I think, yeah, we enjoy people who like to ask questions and like to explore these sort of interesting aspects. And I think the nice thing about host microbe interactions is it can capture a lot. You can have people who like immunology. You can have people who like development. You can have people who are more interested in physiology questions. And so it really does encompass a lot. So I think students who find themselves maybe unsure which way they want to choose, it's something maybe to think about, is that sort of there's a breadth of angles you can take in sort of these host microbe interactions.
I really, you've heard me say it before, just as we talk about wanting a big tent to get as many different faces and voices involved as we can, perhaps a big petri dish is one way to say it. And I hesitate to think of how that cartoon would look, but it's certainly important. What I'm really taken with about your work is there's a level of it, and I don't mean to minimize it, is something that an absolutely new student could get involved with. And at the other end are the more high-tech things such as RNA-seq that you can do, where you can determine specifically what genes are being expressed at particular times.
Absolutely. I have, and I have a lab and I was trained in labs like myself that allowed a lot of undergraduates in. And so I have, in fact, a number of first-year students who are going to be picking up on some of these, exploring, if we add different microbes and expand sort of these interaction assays and that I talked about in the first part. Because to me, that's a totally approachable project to give a student, even if they've never worked with a microbe before. And so we're excited to see what they're going to be doing over intersession.
And it's so interesting. Lactobacillus, on the one hand, part of a lot of fermented foods, Acidibacter, necessary to make the very vinegar that you put in your vinaigrette.
Exactly.
So I used to really enjoy teaching folks about that. So, Nichole, I like to ask a question of people as follows. What is the one observation, the one experience that sold you as a microbiologist?
Well, it just so happens that you brought up the serratia, and that was actually my first exposure. It was the charismatic microbe of serratia, and I remember as an undergrad, in my first micro experience, working in a lab and having the master student who was showing me techniques turn to me and say, "Oh my gosh, look at this microbe! It's so interesting how it grows." I think she gave me a plate wrapped in parafilm to take back to my dorm room, and I watched it grow like over the plate, and then brought it in the next time we met. And so it was a serratia strain, that a prodigiosin producing strain. Although this one I think did the variation and the phenotypic switching, so I got to see the white colonies that came out of it too. But I think that was the first thing where I was like, "Wow, this is a fascinating organism to watch. It's got this amazing color," and that kind of got me hooked on microbes being cool.
You just pushed my happy button when you talked about the phenotypic switching, because I've been looking into that myself, and we think we know some things, and I will tell you another time about it. And I'm very excited by it. I
Fantastic.
don't have your kind of tools, but I do have a brain.
That's all it takes.
It's true. Well, at that point, I'd like to kind of start thinking about ending the segment. So can I ask if there are any take-home lessons? That's an expression I use all the time, and sometimes my students said, "What does that mean?" So overarching take-home lessons from today's presentation you would like people to remember?
I think that we can use simple systems and models to understand complex biology.
That's certainly a very, very important thing. I only bring this up because I often run into people who remind me very much of my late parents, who will say, "Well, why is it that you look at these things?" And I think the ability to be able to put that in a way that's accessible is so important. And you've certainly given examples of all that. I love the way that we've worked in organisms that we know of that are involved with human health, that are involved in some things that take place in your model system of
Mm-hmm.
Drosophila. Uh, this very interesting pathogen, I'm going to call it a pathogen. I know that that word is not popular at your home.
Oh, every time it came out of my mouth, I was like, ah.
Well, that's because the term pathogen doesn't refer to the host side of things. This is one of the
Exactly.
things. It's all context, 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 much as the way a real estate agent says location, location, location.
Exactly.
But it's still that, I'm going to say it, that pathogen can affect gut development, and specifically the leaky gut situation.
Mhm.
I can't help but think that that's relevant to things that people can see in the news.
Oh,
And
absolutely.
of course, making wonderful opportunities for coworkers at all different levels, from walking in the door from classes all the way up to postdoctoral fellows. I think it's a heck of a career, Nichole.
Well, it's a lot of, we get to have a lot of fun, and we're really lucky. And so I think that's the other take home message, is that when you have interesting biology to look at and explore and great people to do it with, it really makes the work that you do rewarding.
One further thing that's been in the news recently is the concept of how often people have to work in a laboratory. There's been a lot of thought about work-life balance.
Mhm.
And one thing that I really say, it doesn't have to do with the number of hours, because I think some folks misunderstand that. The drive to know what happens next. when you do an experiment, when you streak a plate, you want to know what it looks like the next day. And that doesn't have a whole lot to do with how many hours you put into it. It has a whole lot to do with the burning interest that you have yourself.
Absolutely. Yeah.
And for me, that's the goal. And it sounds like you share it.
Absolutely. No, I think those, you can do efficient science and make great discoveries. And so I think that's something to keep in mind. But knowing the next question or just what excites you and what you think is interesting, and there's no right or wrong answers there. But I think then having that interest and inquisitiveness to sort of keep going the next step, because a lot of what we do and most of what we do is small steps to get to the big discoveries. And so
we call it the and research.
Exactly.
Thank you so much for joining me today on the podcast, Nichole. It's always such a pleasure to see you.
Well, it was really great to see you, Mark, and to share with your listeners. And I'm enjoying this series and can't wait to hear others.
Well, you're very kind. My very best to you and yours.
Likewise.
This has been Matters Microbial, a weekly podcast about the wonders of microbiology and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, including wonderful links as always, can be found at microbe.tv/mm. If you like what we do here, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Nichole Broderick can be found in the biology department at Johns Hopkins University in Baltimore, Maryland. Many, many thanks to David Renata for excellent editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum today. See you next week on Matters Microbial.